All right, let's talk about Chapter 17, Evolution and Diversity of Animals. Um, a lot of animals people don't consider animals. Most people, when you say animals, think you're talking about stuff like raccoons and owls, basically the vertebrates. But there are a lot of animals that do exist that are not vertebrate, and we're going to be covering some of those. Here's a couple of things. To be an animal, these are our characteristics. You have to be multicellular. Single-celled organisms are no longer considered animals. There was a time when the protists were included as animals, but no longer. You have to have eukaryotic cells that lack cell walls, meaning we're eukaryotes. We have a defined nucleus and nuclear membrane. We are heterotrophic. We eat and take in energy by ingesting food. Our embryo development is a hollow ball stage, has a blastula. Okay, so at some point you start out as a single cell when the sperm fuses to the egg, and then you undergo this mitosis. Basically, you just to 2 cells, to 4 cells. Eventually, you form a big ball of cells called a morula, but then you will go into the blastula stage in which you will form a hollow ball. Now, the cavity inside this blastula stage will become your coelomic cavity. Your cells will bind to extracellular matrix, so all of your cells are attached to things and held in place. Some of the first animals arose from aquatic protists. Looks like we see some of the first animals showing up in the fossil record about 570 million years ago. And several different types of animals, but these were not very sophisticated. Then here in 543, we had what was called the Cambrian explosion. That's when we just started seeing all kinds of multicellular animals. First vertebrates on land. We didn't see the oldest fossils are 375 million years ago. Then we start seeing the rising of the modern groups, the reptiles, the mammals, etc. Fossil record shows the first Homo sapiens, that's our species, showing up about 200,000 years ago. Now, there were other hominids, and we actually competed with several species of hominids. We're in the family Hominidae, but we outcompeted them. So they went extinct, and we survived. We won the natural selection battle, I guess you would say. KT extinction event. Extinction events occur for multiple reasons, but about 65 million years ago, in the Yucatan Peninsula, that's part of Mexico, okay, there was a huge meteor strike, okay, and when a meteor pounds in to the Earth, several things affect its seriousness. If it comes in at an angle and kind of skims across the Earth, it's not as bad. But when it comes in at a right angle and just shudders the Earth, that is when you get the most what is called ejecta. So this thing plows into the Earth, and as it does, because this meteor is going at exceptionally high speeds. Now, there were a couple of movies. It's been a while now. One was called Armageddon with Bruce Willis and Liv Tyler and Ben Affleck. And another one was called Deep Impact. And in both of those movies, there were people standing out watching pieces of the meteor coming down. But that's not how it would really happen. As the meteor enters our atmosphere, it would start to glow from the friction. It would glow red hot, then blue hot. Eventually, the meteor would be violet hot. It would be giving off so much heat that if you were observing it, it would vaporize you. Okay? It would burn and sear and scar the landscape anywhere it could be seen. That's how much heat it would be giving off. And then when it hits, it explodes. And it digs a crater. And all that dirt and stuff in the crater gets thrown up into the air. And in the air, it's called ejecta. Okay? And so much of this dirt and ejecta and thing was shot into the air that it blocked the sun with this giant dust cloud that surrounded the planet. This inhibited photosynthesis. Plants started dying. They were unable to reproduce. They went dormant. The herbivores started dying. And it just caused a chain reaction that wiped out the majority of the what are called macrofauna. Macrofauna are the big animals. Okay, some of the smaller animals could survive, but they did take population hits. Now, we had basically an indication that this occurred. Okay, and the indication was that there was this iridium layer between the Cretaceous and the Tertiary. All right. Now, the Cretaceous is called KT because the first people to name the Cretaceous were the Germans, and they used K instead of C for the K sound. And the Tertiary has now been changed, and I can't, off the top of my head, I can't think what they changed it to. But anywhere you could access rocks that were from the Cretaceous era, and they met the Tertiary era, which is our modern era, you would find a layer of iridium. Now, why is that important? Well, iridium is only found in meteors. We don't have any natural deposits on this planet. All the iridium that we use and find come from previous strikes of meteors. All right? And iridium is very valuable and is used in several different manufacturing processes, so it is mined and refined. And here is that iridium layer. So what that told us, since we found this layer everywhere on the planet that we could access these rocks, that implies that there was a dust cloud of iridium that covered the planet. Okay, sort of like a couple inch thick layer of snow. And it takes a while for iridium, because it's a very small molecule, to settle out of the atmosphere and form these layers. Long enough to interrupt the photosynthetic process and damage the plant life and then the herbivores. Herbivores are animals that eat plants. But we couldn't find the crater. Well, the reason we couldn't find the crater is we weren't high enough. Once we started getting satellite imagery, this is how big that crater was. It's in the Gulf of Mexico. And here's the Yucatan Peninsula, and there's Mexico, and then there's, I think, maybe Honduras is right there, if I'm not mistaken. And so the first time we found this crater was from space, and here are some of the actual satellite shots showing the crater. All right. There's Doppler radar doing a 3D imaging of the crater. It has since filled in. This area here is the effluent out of a river system, and so silt has built up there. But that gave us the data now to confirm what the layer of iridium around the planet was implying, that a huge, huge crater was made by a huge meteor, and that caused 70% of the species on the planet to go extinct. They just stopped at that point. They never, the fossil record, they just ended. Okay? And they've drilled into this crater to try to find information. Okay? And we've been testing this and working with it. Okay? And it has provided a high yield of important information. I'm not going to go into that here. That's really not the purview of this course. But this crater has been confirmed as a meteor crater. Okay, that's what's important there. Now, this is called a cladogram. This is how we classify animals based on genetic similarities. So this is the evolutionary tree for all the different animals. Now, ancestral protists and multicellularity. We have protists that are still alive. We have protist colonies, like Volvox, that basically show the first case of a colony of organisms coming together, and there's a division of labor. Okay? And understanding that origin of life is still a hypothesis. It's not a theory. However, evolution is a theory because we have enough data to support that, and that's primarily through genetics now. All right? And there are some important parts in our evolution. At some point, this ancestral protist became multicellular. We have quite a few examples of living early multicellular type of animals that would have led. At another point, we had tissues arose. These first organisms didn't have tissues. Okay, they just had a conglomeration of cells. And so the most primitive organisms on the planet are these sponges. Sponges don't have any true tissues. They don't have any symmetry, etc. We'll talk about sponges. They're all marine. There's a few freshwater sponges, but not many. And when cells started developing tissues, which we will talk about in a future lab, we developed two types of symmetry: radial symmetry and bilateral symmetry. Bilateral symmetry would be like, take a human. We can chop a human in two, but there's only one way to chop them in two to have exactly equal parts. You can cut them into a right half and a left half, and they will be equal. If you cut them into a top half and a bottom half, they're not equal. You cut them, front to back, they're not equal. There's only one way to cut something in two to be bilateral. So anything that has a right side and a left side is going to have bilateral symmetry. Radial symmetry means there's many, many ways to chop something into two equal parts. Imagine a perfectly round pie. Okay, if it's perfectly round, I can cut it into two exactly equal parts. Okay, and the primary groups there are the cnidarians, but the cnidarians have radial symmetry in the medusa or free swimming stage. Sometimes they don't have radial symmetry in their polyp phase. Okay. Another big evolutionary leap were the number of germ layers. There are 3 germ layers: endoderm, ectoderm, and mesoderm. The early animals only had 2 germ layers, endoderm and ectoderm. These are in the fetal stages, and ectoderm and endoderm, basically as it reproduces, will evolve and change into the many different tissues and organs and things we see in a living organism. We see a 3rd layer form in the bilaterally symmetrical, more advanced animals, and this is the mesoderm, which tends to develop into all the very complex organs that we find in animals. Then there's deuterostomes and protostomes. Okay, deuterostomes and protostomes. Here we go. Let's find my deuterostome and protostome group. Here we go. Protostomes, what happens? Here's mesoderm develops later. First you get ectoderm and endoderm, and then you start getting this little blastopore that will eventually become the mouth, and this area here will become the gastrointestinal tract. Some gastrointestinal tracts go all the way through. Some are just a big sac. Basically, the mouth and the anus are the same. In protostomes, the mouth formed first. Then we in the more advanced animals, this became the anus formed first. And they finally tracked that down and found where the genetic mutation occurred to flip that. Okay, and so we now know a little bit more about our ancient development. So here's our protostomes, mouth first, and all of these groups of animals, all the flatworms, the mollusks. Now mollusks are like snails and octopi. They would also be squid, things like that. Then there's the annelids, or all the earthworms. Then there's the parasitic roundworms. And then, of course, all the arthropods. The arthropods, as far as biomass is concerned and number of species, are the dominant group on this planet still. Okay. Deuterostomes developed a little later. There was this branching, and we developed into the echinoderms, which are things like sea stars and sand dollars, and then the chordates. The chordates include all the vertebrates, etc. But it also includes some groups of non-bony animals like sharks, which are cartilaginous skeletons only. And then there's even some jawless fish that are still around, just a couple of species. And we'll talk about the development of the jaw later. Okay. Here is a good drawing showing bilateral versus radial symmetry. Here we have right and left halves of this lobster or crayfish. I'm not sure which it is. And here we can see if you can divide it into multiple ways in which there's two mirror images or totally equal parts, that's radial symmetry. Okay. The embryonic ball of cells is called a gastrula. Okay, it evolves from the morula into the gastrula, and towards the end of lab we'll go into all the stages and talk about the different stages of embryological development. The body cavity, organization of the digestive tract, segmentation, characterize animals. For instance, annelids, you can look at annelids, you'll see little body segmentations. The coelom in some animals is very simple. In the more advanced animals, it's very complex. Okay, and here are your definitions. Okay, in a body cavity surrounded on all sides by mesoderm, that's a coelom. That's a true coelom. A pseudocoelom has one side by mesoderm and then one side by endoderm. All right, and then some bilaterally symmetrical animals just lack a body cavity. Okay, they just don't exist. So that was an important splitting point. Digestive tracts. They can be complete, like in this human, having a mouth and an anus, and everything flows through. It goes from the esophagus down to the stomach, into the small intestines, out through the large intestines, then out through the rectum and the anus. And then that's called a complete digestive tract. And incomplete is the mouth and the anus are the same thing. Here is a primitive organism that is a cnidarian. Okay, it's a hydra. And food is taken into this cavity, digested, and then the leftover waste is just expelled. Okay, so it doesn't go all the way through. Examples of segmented bodies, there are segmented millipedes, segmented centipedes. Millipedes are harmless. Centipedes are a little more dangerous. Okay, not something, some people handle them. I don't recommend it. But the thing to remember is they've got just little segmented bodies. Okay, these are extreme segmentations. So in each phylum over here, because that's what we're listed, you know, these are the poriferans, the cnidarians, platyhelminthes, mollusca, annelida, nematodes, arthropoda, echinodermata, and chordata. These are all our phylums. And we can trace genetically the arising of these different types of structures. Somebody always asks me, Disozoans has to do with shedding of an external skin in a molt. Okay, so as a crayfish grows, it will eventually outgrow the hard shell around it. So then it sheds that shell. That's called ecdysis, and it's really soft and vulnerable for a short amount of time before it hardens a new shell. Okay. If you've ever had a pet hermit crab, you have to have extra shells because as they get bigger, they'll shed their external shell and take up a bigger shell as a home. Lophotrochozoans, this has to do with mouth parts and the structure of the mouth, which we're not going to go into here. Sponges are an example, have no differentiated tissue. Okay, they're asymmetrical, meaning there's no way to cut them into two equal halves. Cnidarians, radially symmetrical. They have stinging cells for defense, and then they have a free. Most species have a free swimming stage. There are some examples that don't. The flatworms, the most common one you might see would be these little guys here. These little flatworms are called planaria. The mollusks are all your snails. These are called chitons, scallops, slugs, octopi, and squid. Segmented worms, if you go fishing, right, there's earthworms and red wigglers are segmented worms. Leeches are in that group. Polychaetes are a segmented worm found in saltwater or marine environments. Nematodes, these are your unsegmented worms, and they're almost all parasitic. The arthropods have a couple of different types of mouth parts that help separate the two groups. And there's different little body parts and segments on these guys as well. These are pretty advanced. Arthropods include everything from crabs and lobsters to spiders and insects. Trilobites are extinct marine arthropods. You know, I've got some fossil arthropods as well. Very diverse animals, the arthropods. This is a meristomatid crab, or what do they call them? Horseshoe crabs. Ticks and spiders and scorpions are all your different chelicerates or arachnids. Right, millipedes and centipedes are their own little group. Crabs, shrimp, and lobsters. Then we get to the echinoderms. These have spiny skins, and in addition, there's sea dollars, sand dollars, sea stars. These are sea urchins, sea cucumbers. And then we get chordates and the vertebra. Okay? The most primitive chordates out there are these little guys here. They don't have jaws. They're called lancelets. And they have these little gill slits, dorsal nerve cord, a notochord, and a tail. To be in chordate, at some point in your life stages, you have all four of these things. Even humans. We have primitive gills as an embryo. We have a tail as an embryo. We have a dorsal nerve cord and a notochord as an embryo. The notochord becomes our spinal column. The nerve cord the notochord becomes the backbone, and the dorsal nerve cord becomes your spinal column, all of the spines in that area. Those are the four major characteristics of vertebrates. Here's a primate skeleton. The cranium and the vertebrae and the rib cage is called the axial skeleton, and then the pectoral and pelvic girdles with the legs are called the appendicular skeleton. Here they are, the four major features of a chordate. And if we split our chordates out, we can put them up here and compare them. The tunicates and the lancelets are obviously the most primitive. The hagfishes and lamprey, these animals don't have any jaws. Then we have cartilaginous fishes like sharks and guitarfish. You get the bony fishes. These are the ones you're probably most familiar with. Amphibians. You have reptiles and birds and the mammals. And the birds have now been given a little sub-branch. They are now called avian dinosaurs because the dinosaurs are now classified as non-avian dinosaurs. Here's tunicates. These are pretty, and lancelets. Lancelets just bury themselves. You hardly ever see them. You have these little tunicates, and they're filter feeders, but they do have a notochord. Okay, they just never get a skeleton. Here's a hagfish. They secrete this nasty mucus. There's a lamprey, and they have no jaw. They just have a sucker. Here's a coelacanth, the bony fish. This was thought to be extinct, and the fossil record shows it dying out at 65 million years ago, but then we found some small populations in very isolated places in the world. Some have been found in Indonesia now, and some in the trench between Madagascar and Africa, but they are on the decline. Interesting fish. In the 1920s, they were rediscovered by a fisherman fishing between Madagascar and Africa. And he pulled one of these things up, and he's like, wow, what the heck is this? Because he was deep-sea fishing. And they took it to the French naturalist, because at the time Madagascar was a French colony. And he identified it as basically a fossil fish that they thought had went extinct. Then the amphibians, you have several types. Caecilians look like little worms, but they aren't. All right, they are a very primitive amphibian. Rubber eels are a good example of those. You can buy those in some aquarium places. Salamanders and frogs, toads, things like that. The amniotic egg broke the tide of water. Up until that point, these guys still go back into the water to breed. That's what amphibians mean. It means dual life. You have a life stage that lives on land and a life stage that lives in the water. Amniotic egg, basically they could lay an egg and it would develop a hard shell so that it wouldn't dry out. Okay, and so animals started congregating on land and spreading. And here's some of the first vertebrates on dry land were reptiles. Huge group of those, and then they started developing. A branch broke off and became dinosaurs, and those dinosaurs evolved into birds. Eventually, the mammals showed up. Now, there's some very primitive mammals called monotremes, and the echidna, they basically lay eggs, and when the eggs hatch, the young, they just have little pores, and the young just lick the milk off of the pores that are secreting milk for them. You have the marsupials, in which the young are born very, very primitive and crawl out of the female and crawl into a pouch and latch onto a teat and develop in the pouch, and eventually coming out. The only marsupial we have in the United States is this guy, the little opossum, kangaroos. And then you have the more advanced mammals that basically are called placental, in which they carry their young inside, fed by a placenta, and are born alive. Okay. Human evolution. We are placental mammals. We are a member of the group of primates. All right. Here is kind of the way it breaks down. Some of the common chimps and bonobos, these are two different groups of chimpanzee, are the most genetically similar to us. Out of 20,000 plus gene sites, the difference between the bonobos and the chimps and us is only about 1.5% of that, which I think comes down to about 300 genes are different. The rest are exactly the same. Okay. Shows some of these different groups. Now, hominids are the great apes and include the orangutans. Okay? The hominoids are the apes and include the gibbons. And then the hominins are us, the chimps, and the gorillas. Okay? These are important distinctions. Fossils and DNA tell the evolutionary story. The difference between these guys and us, one of them is the shape of the backbone. Now earlier, if I can find it, here's your typical primate. They always have this little bowed or arched backbone because that facilitates them walking on four legs 99% of the time. Okay, they can rise up on their hind legs, but that's not they walk very awkwardly. There is in the fossil record a group of primates that did evolve a different pelvic structure and the straight backbone, which was a step towards what is called bipedalism. Okay. And there were advantages to being bipedal, which obviously were selected for. Here is the migration. There's a couple of various hypotheses on how we migrated. Everybody pretty much agrees that the first humans arose in Africa. Okay. We have found no hominids that are older than the ones found in Africa, and some people think there was one major migration out of Africa, and some people think there have been multiple migrations of humans. How did they get into North America? Well, during an ice age, this was called the Bering Land Bridge, and they would have came from here in the Far East, traveled across that, and colonized. Now, we've recently had some finds showing that humans migrated to these regions much earlier than we thought. Okay. Fishapod, transitional species. This is a very interesting critters. These are ones who show both fish-like and amphibian-like characteristics. Okay, Acanthostega and Ichthyostega, but they still look a lot like amphibians, but not completely like them. Then there's the fishapod. This was found in rocks 375 million years old up in the Canadian Arctic. And in the Canadian Arctic, that's where they should have been. Basically, looking at fossils, we can figure out, okay, if there is a transitional species between fish and amphibians, based on the fossil record, it should fall in this realm of rocks and how old they are. And then we can look at that, and basically a man named Neil Shubin found two areas where you could get to rocks this age. One was in Pennsylvania, which did not yield any of these, and the other was up in the Canadian Arctic, and he found the fishapod. It's called Tiktaalik. It was a tremendous paleontological find. Basically, if you look at it here, the front half is flattened like an amphibian, the back half is flattened like a fish. Front half has very primitive flipper-like legs. The back half just has fins. So, there you go. That wraps up this chapter. Thanks a lot.